to acknowledge the previous talk as a really outstanding background for now. About half of my background slides will be obsolete, which will make it go a lot faster. The one significant difference, I think, is that the work that I'll talk about is entirely in mouse, so disregard any of the human references to the background, and everything else is the same. In my lab, we've been interested for a number of years in understanding how antigen presentation occurs in both helpful and autoimmune immune responses, and in particular, the role of transcription factors in driving T cells and antigen presenting cells towards a phenotype that generates autoimmune diseases. So again, pretty much this will be redundant background, but just to orient you towards what I'll talk about in thinking about the kind of requirements for an autoimmune response. What we think about in terms of targets and how these autoimmune responses. Antigen presenting cells have to take up antigen, become activated, and activate T and or B cells of a CD4 and or CD8 nature for the T cells. And then those T cells go on to destroy, in the case of MS and EAE myelin. What we're interested in this particular portion of my lab is how T cells, in however you want to divide them, turn into the T cell phenotypes that create these harmful autoimmune responses. So transcription factors, in particular, are driving factors for polarizing naive T cells towards Th1, Th2, Th17, Tregs, and there are many subsets which were very nicely outlined in the previous talk. So one of the things that my lab has been working on is a transcription factor called Kruppel-like factor 4, or KLF4, which belongs to a family of zinc finger transcription factors. It's probably better known for its role in maintaining stem cell function. There were studies showing that KLF4 is one of the transcription factors that, when put into mature fibroblasts, confers an embryonic pluripotent stem cell phenotype. What we got interested in this factor for was some results that came out of Kurt Siven's lab, who at the time was my next door neighbor, and John Aldero's graduate student, who discovered that the KLF4 was required for the differentiation of this inflammatory monocyte cell phenotype, which they defined as a GR1 CD115 positive cell. And we kind of had started discussions about whether KLF4 would have a role in the autoimmune response based on the requirement for some of these monocyte type cells in the differentiation of autoimmune responses, in particular EAE. So we first just sort of globally assessed whether KLF4 was required for the generation of EAE. And in inducing EAE, the mice are immunized with the myelin basic protein, which leads to activation of T cells, destruction of myelin, and the activated T cells essentially traffic into the CNS, where they again meet up with antigen presenting cells and destroy myelin. So what we're interested in is whether these CD115 GR1 inflammatory monocytes were potentially necessary for the destruction of myelin, or whether they also played a role in activating T cells within the CNS. So what the results of the first sort of global study turned out is that the mice that were deficient in KLF4 were essentially resistant to the induction of EAE. So we next asked why. In the process of the active immunization, the mice are immunized with an antigen, an adjuvant, and both dendritic cells are required in that they have to take up the antigen, process it, present it, and then activated T cells go on to cause disease. So we first wanted to assess whether the absence of KLF4 in either the dendritic cells or in the T cells was necessary. So we first analyzed the phenotype of the immune systems in the KLF4 knockout and compared to wild type, thinking that perhaps there was some defect in the ability of T cells to activate or differentiate or traffic to the CNS. And what we found was that both CD8 and CD4 positive T cells in the spleen, that there was essentially no difference in the T cell development, equivalent numbers of T cells developed and were present. But before and after immunization, looking at CD44, CD25, there was no real difference in the T cells from the KLF4 deficient mice, indicating that KLF4 wasn't required for the development of T cells in the thymus or migration to the spleen, development of the immune system. In contrast, what we found was that the number of CD11B positive cells were significantly decreased in the KLF4 knockout mice in both the brains and the spleens. This, which was pretty consistent with the notion of this being important for inflammatory monocytes. So what we asked was, what's the role of KLF4 in the induction of active EAE in T cells, inflammatory monocytes, and dendritic cells? Similar to what was found in the resting state after immunizing mice, we also observed that in the brain there was a decrease in the inflammatory monocytes. After immunization, a number of these sort of GR1 positive, CD115 positive cells migrated to the brain. In addition to the decrease in CD11B cells, we also observed a decrease in dendritic and NK cells in the brain after, and brain and spleen after immunization, but no difference in T cells. And this was true both, again, in the number in the spleen and also in the number of T cells of both CD8 and CD4 that migrated to the brain. So at this point, we actually really had no idea what the difference was between the KLF4 knockout mice and the wild type in terms of why they had such a broad resistance to EAE. And we were further puzzled when we discovered that KLF4 deficient dendritic cells were just as effective at stimulating T cells if we took in vitro, took out CD11C cells from either wild type or knockout cells, put them in culture with 2D2 cells, added MOG. They proliferated. Using either purified dendritic cells, using whole spleen, regardless of the purity of the cells, there was no difference in terms of their ability to stimulate T cells. And when we looked at whether there was a difference in the ability of the KLF4 deficient mice to develop into memory T cells, again, there was no difference, indicating that kind of long-term that they were able to maintain whatever memory phenotype they had developed. So, sorry. So in order to look a little bit more carefully at whether there was something about the KLF4 knockout mice that suppressed their ability to activate MOG-reactive T cells, we adoptively transferred the MOG-reactive 2D2 transgenic T cells that are specific for the myelin oligoprotein, transferred 2D2 cells into either wild type or knockout mice, immunized the mice, and looked at the ability of the wild type or knockout T cells to lead to expansion of the transgenic T cells, which removes sort of the variable of that both the APCs and the T cells were knockout in the previous experiment. And after transferring the 2D2 T cells immunizing and analyzing the number of 2D2 cells that became activated, migrated to the brain, there was still no difference in either the wild type or the knockout mice. In addition, the CD11B cells that we looked at in the brain had equivalent abilities to take up antigen. We thought that it was possible that cells were maybe possibly deficient in their ability to phagocytose antigen, but essentially there was no difference in those. So the next question that we asked is, how is it possible with all these features being the same that these mice are still so resistant to induction of EAE? So we next asked whether we could bypass the resistance by providing adoptively transferred pre-activated T cells. So in this case, rather than asking the KLF4 knockout mice to take up and process and present antigen, that we would utilize pre-activated MOG reactive T cells to induce disease. And the schematic for essentially how this experiment occurred was donor wild type mice are immunized with MOG, which generates a large number of T cells that are specific for MOG. And then those cells are taken out, cultured with MOG, and injected into recipient mice, which were either wild type or KLF4 knockout, and then we evaluated the onset and progression of disease. And using the adoptive transfer of wild type MOG reactive T cells, the KLF4 deficient mice were still resistant to the induction of EAE. So in the absence of activating, the need for activating T cells kind of in place, we wanted to next assess what cells were preventing the disease, or whether there was a further activation that was required that wasn't taking place in the KLF4 knockout mice. So in order to assess what was happening from the adoptively transferred cells that were wild type MOG reactive versus the host cells that were either wild type or KLF4 deficient, we utilized a chimeric model in which we immunized the wild type mice for donor cells. We immunized CD45.1 mice to generate CD45.1 MOG reactive T cells, and then transferred those into CD45.2 wild type or CD45.2 KLF4 knockout, so that we could assess what the difference was between the pre-activated adoptively transferred cells and the host. And then by FACS, we were able to separate CD4 by CD45.1. So the host being CD45.2 and the adoptively transferred pathogenic cells being CD45.1. And what we found was that the transferred CD45.1 MOG reactive cells, when transferred into wild type hosts, made gamma interferon IL-17, and when transferred into wild type hosts, the host also made gamma interferon and IL-17, but if we took wild type MOG reactive CD45.1 cells and transferred them into KLF4 deficient T cells, while they made equivalent amounts of gamma interferon, the host CD45.2 KLF4 deficient T cells made no IL-17, and in fact had a suppressive effect on the transferred in CD45.1 T cells in their ability to make IL-17. So essentially the transfer of the MOG reactive wild type cells into wild type mice led to an activation of the host the host CD4 cells to make both gamma interferon and IL-17 in kind of this infectious process. And then, but when they're transferred into the wild type, the host T cells were unable to make IL-17, and in fact, there was less IL-17 in the transferred cells. So that was our first indication there was something really significantly different about the T cells that were deficient in KLF4. And these results are from the spleen. While we had a lot fewer cells, it also turned out that that was true in the brain as well, that in wild type IL-17 producing T cells traffic to the brain, whereas in the KLF4 deficient T cells, deficient hosts, they didn't. So we wanted to sort of set forth this as a hypothesis that there was the trafficking of these KLF4, or the lack of the trafficking of the KLF4 deficient T cells into the brain that helped to protect the CNS tissue from the damage done by the normal induction of EAE. So we still weren't sure, since there was a requirement of the host CD45.2 cells to actually activate the T cells. We wanted to ask whether there was something intrinsic to the T cells that KLF4 was required for the induction of IL-17, or whether because the APCs, dendritic cells, antigen-presenting cells, were also deficient in KLF4, whether the loss of KLF4 in the cells that drive the T cells towards Th17, whether those were, whether that also led to the inability of T cells to make IL-17. So we took out purified CD4 T cells and naive CD4 positive T cells, and used artificial stimulation in vitro using anti-CD3, anti-CD28 stimulation with cytokines polarizing towards Th1, Th17, or Tregs, and compared the ability of the wild type CD4 T cells and the knockout CD4 T cells to polarize towards gamma interferon secreting, IL-17 secreting, or FOXP3 expressing T cells. And again, what we found was that there was essentially no difference in polarizing towards gamma interferon secreting cells or FOXP3 producing cells, but where wild type KLF4 T cells polarize very efficiently towards Th17, there's essentially no polarization towards Th17 in the KLF4 knockouts. So this led us to think that there was a intrinsic role for KLF4 within the T cells themselves, that KLF4 might be required for the induction of IL-17. So while it was required for polarization, one of the sort of interesting features was that there's no difference in CD4 T cell proliferation, and there was also no difference in the Th17 Treg axis. So there was no compensation for the loss of Th17. Essentially, these cells seem to stay in an undifferentiated, though proliferative state. So we next asked, because of the sort of directness of the requirement of KLF4 for IL-17 production, whether since KLF4 is primarily a DNA binding transcription factor, whether it had a direct role for the regulation of IL-17, meaning was it required, did it bind to the IL-17 promoter, and was it required for IL-17 expression? So we used a chromatin immunoprecipitation assay to assess whether KLF4 actually bound to the IL-17 promoter. There are two potential binding sites for KLF4 within the promoter, and using two different antibodies to immunoprecipitate KLF4, we found that KLF4 did indeed bind to the IL-17 promoter. We've since actually gone on to conducted luciferase reporter assays and shown that it not only binds, but it also activates. So we started thinking about the role of KLF4 in this particular differentiation of naive T cells towards the Th17 and the kind of canonical transcription factor that's generally considered to be required for Th17 and mice ROR gamma T, and thought that perhaps there was an effect of KLF4 on the expression of ROR gamma T. But when we looked by messenger RNA, comparing the induction of ROR gamma T in the process of polarizing naive T cells towards Th17, there's very significant upregulation of the expression of ROR gamma T in both wild type in the first and third bars, and in knockouts as well. There's essentially no difference in the level of messenger RNA for ROR gamma T. So it looks as though KLF4 kind of acts either downstream or independently of ROR gamma T. So although we found that that it had this intrinsic role within the Th17 cells and the requirement for the induction of IL-17, because we had seen a number of possible differences in the sort of activation as well as the numbers of antigen-presenting cells, we next wanted to evaluate whether KLF4 in vivo functioned both at the level of the T cells themselves and also the number of antigen-presenting cell factors that lead to T cell polarization. So in particular, IL-6 and TGF-beta, and also GM-CSF, which isn't necessarily thought of as a polarizing, but a necessary for dendritic cell function. So the cytokine that is sort of particularly thought of in terms of polarizing and maintaining Th17 responses, IL-6 was what we analyzed first, in particular getting back to the EAE studies where we found that the KLF4 mice were essentially resistant to EAE. And the IL-17 knockout mice, while they have delayed progression and somewhat resistance, didn't have as significant a level of resistance. So in trying to explain why the mice were pretty much entirely resistant to EAE, one of the clues in the differences with the differences in the antigen-presenting cells, and mice that are deficient in IL-6 are pretty resistant to EAE. So what we had found when we compared the levels of secretion of IL-6 from the KLF4 knockout and wild type dendritic cells is that the DCs that were deficient in KLF4 were essentially secreted no IL-6. So they had as great a deficiency in the secretion of IL-6 as the T cells did in IL-17. And again, we wanted to look at whether this was a direct function of the transcription factor function of KLF4. So we analyzed whether the KLF4 bound directly to the IL-6 promoter, again by the chromatin immunoprecipitation, and it indeed does bind directly to the IL-6 promoter, indicating that it has a dual sort of regulatory function on both the secretion of IL-6 and IL-17. And I'll just go through the last couple slides quickly because I think I'm out of time. But to make a long story short, it has no effect on IL-23 and no effect on GM-CSF, where it does. It has not nearly a significant effect on the secretion of TGF-beta, but the cells that are deficient in KLF4 had about a 50% reduction in their secretion of TGF-beta. So our ongoing hypothesis is that KLF4 acts as a molecular switch that is necessary to generate the autoimmune and or inflammatory responses. It doesn't have any noticeable regulatory effect on T cells in a homeostatic state, but it does regulate the generation of macrophages of this GR1 CD115 high. And primarily, what we are working on with this hypothesis is that the activity of KLF4 and the binding of KLF4 onto the promoters of some of these critical cytokines in both dendritic cells and T cells helps to provide this coordinated regulation of both the dendritic cell and the T cell side of the generation of this autoimmune response. And I meant to acknowledge people as I went along, but Lori Lebson, Jason Rosenzweig, and Ann Goki are kind of the primary people who have conducted essentially all of the experiments that I showed you, and John Alder and Kurt Sivan, who have been ongoing collaborators since their discovery of the lack of inflammatory monocytes in KLF4, and Peter Calabrese and lab for their expertise in EAE and MS, and Jonathan Powell and Chris Gamper, who primarily got us oriented towards the in vitro polarization of T cell process. And I'll stop there.
We have time for a couple of questions for Dr. Wortley.
Is there a concern that sequence for the promoter of IL-6 and IL-17 will CLF4 bind to?
Oh, yeah, sorry. The question was, is there a consensus sequence for IL-6 and IL-17? And the answer is yes, there is. And interestingly, there is not one in IL-23, and we don't see any difference there. So we think that that it does need to bind to its consensus sequence. Although one of the questions is whether it acts as part of a kind of complex that may use alternate sequences.
Thank you very much for a very interesting